Alright, so in this video, I'm gonna be talking about the proteins in DNA replication. Before I start, I just wanna let you guys know that we've posted videos about DNA replication itself, like just the concept of it. I would suggest watching it first so that you would understand this better. I think it would be really helpful. Both me and Kenzie posted our own videos, so you can find it in the canvas. So yeah, I guess let's start. So in biology, every time you can almost always assume this. Every time that you see an ace, like a word that ends with an ace or something like this, primase, polymerase, topoisomerase, helicase, you can always assume that it's an enzyme. And enzymes are really important because they catalyze a lot of reactions. But for this one, we're just gonna focus on DNA replication. All right, before I go and try to incorporate it in actual DNA replication, I'm just going to talk about the function really quickly. So, first of all, helicase. Helicase basically unzips the DNA double helix. One way for me to remember that is because I know that DNA is a double helix. And what deals with a double helix? Basically a helicase. The heli in both helix and helicase is what helps me remember it. Next one is topoisomerase. Topoisomerase basically relieves or prevents supercoiling. If you were in class, Dr. Zolman showed you guys what supercoiling means and like what actually happens and why it happens. So for me, I don't have any kind of way to actually visualize it, so I'll just use this one. Imagine this is the DNA, right? And imagine helicase is going that way to unzip it. So as helicase goes, it actually has a force that pushes the DNA. Without topoisomerase, what can happen is that I keep pushing and pushing until it starts coiling like this, right? And we wouldn't want that, right? Because we're not going to be able to replicate our DNA correctly. So basically what happens is topoisomerase will come around on the back and make a cut on it, so like it relieves the tension when helicase is pushing and also like making its unzipping motion. So when topoisomerase is present, it's basically like just allowing it to be less tension, and then it will not supercoil. I don't know if this helps, but I hope it can help you visualize what I actually meant about it. So yeah, that's what topoisomerase is. Next one is RNA primase. This is an important enzyme because it makes the RNA primer. So why is it important? RNA primers, we need RNA primers for DNA polymerase to know where to start. Because DNA polymerase, it will do everything else, like it will elongate the new strand that is forming, but it cannot start on its own without the RNA primer. The RNA primer is basically a sequence that tells the DNA, hey, come here, start over here, and this is where you're gonna start replicating your DNA. So yeah, RNA primase, if it's an enzyme, we're talking about ACE, so primase, RNA primase, it makes the RNA primer. That's how I remember it. RNA primase makes the RNA primer. Next one, DNA polymerase 3. This is basically like the enzyme that elongates the daughter strand that is forming. It elongates from 5' to 3'. You won't need to know why it does that, but it has something to do with its proofreading ability and also like how the energy is used when phosphates are removed from the nucleotides. So yeah, DNA polymerase 3, basically the main enzyme that elongates the new strand. DNA polymerase 1, it can do the same thing as a polymerase, any DNA polymerase 2, but its main function, it removes the RNA primer. Because remember, we cannot start without the RNA primer, but in the final product, we don't want the RNA sequence there. We want a DNA sequence. So basically, DNA polymerase 1 will go back, remove that primer, and then replace it with a DNA sequence. So that way, in the final product, we have just one long DNA strand, right? And then last but not the least is single-stranded binding protein, or I'm just gonna shorten it with SSBP. It's basically proteins that bind to a single strand of DNA. And why is it important? Because when helicase unzips the DNA, the DNA itself can form back again. It can start um, connecting back to each other or bonding back. Like the hydrogen bonds will just start bonding again. So basically, single-stranded binding protein helps with preventing that to happen. And for me, best way for me to remember is I read it backwards. Protein binds to a single strand. That's how I remember it. I think some people have different ways of remembering it. Kenzie might have her own way of remembering it too, but this is how I remember it. I hope this helps. And let's go to the next one. Alright, so in this part of the video, I'm just going to be talking about or showing you how the enzymes work in an actual DNA replication. As you can see, I'm only dealing with one side of the replication fork because it would just be the same with the other side as well, but kind of like opposite. So for this one, the replication fork, the direction is going to this way, right? All right, so let's just go over like in order of the enzymes. First of all, helicase. Remember what helicase does? It unzips the DNA double helix. So you can pause the video and then think about like where it would be and then go back again. So if it's unzipping the DNA, helicase would be around here, right? He would be around there and then going to that direction. Because basically these lines over here, these are the hydrogen bonds between the bases. And it's basically like unzipping it so that it will open up like that. Right? Alright, so helicase is done. What's next? Topoisomerase. Topoisomerase, like I said before, it prevents supercoiling. So if it was you, like where do you think topoisomerase would be to prevent supercoiling? So topoisomerase would be around here, right? Because it has to relieve the tension on the part where the coiling could happen, right? Basically, topoisomerase will make a cut on the phosphate backbones just to relieve the tension. So this is where topoisomerase will be doing its job. Like around here, it could be way back in the DNA. But just to visualize it, this is where topoisomerase, will be. Next one, single-stranded binding protein. Remember, like my trick is to read it backwards. So basically, it will bind to a single strand of a DNA. And why does it do that? Because it wants to prevent the DNA from closing up. So you notice that this part of the single strands are open, right? So there's a chance that they can close back up again and bond to each other. And to prevent that, SSBP will come and just bind, like let's say around here. It will just bind around here just to prevent it from closing up, right? So SSBP is done. What's next? RNA primase. RNA primase, like I said before, it is very important for starting the DNA elongation because it makes the primer. I don't know if you can see this, but hopefully you do. I'm just going to use a pencil. Remember, when we're making new strands, it starts from 5' to 3'. So for me, best way for me to do it first is because I know my polarity is here, 3'. That means I can just start over here in 5'. Right? It's the 5' and it will make an RNA primer. I will just label this. Basically, RNA primase will make an primer that is 5' to 3'. Now, what's next? Now that we have a primer, DNA polymerase can now do its job. And the main DNA polymerase that will do that job is DNA polymerase 3. And this is where I want you to incorporate what you've learned so far. Remember, we can only add at the free 3'. So, in this case, there's a free 3' over here, right? So that means DNA polymerase 3 can just add here. From 3', it will just add it and just keep making the strand. Wait. right? So this will be the new 3' of the strand, and that's where DP3 comes in. It's the same way for the lagging strands. If you still don't understand how lagging strands work, I would suggest watching the videos that we posted or reviewing your notes. So basically, lagging strands will have their own RNA primers. Let's say these are the RNA primers, and we know that in the polarity, this is the 5', so that means it should be the opposite. This should be the 5' over here, 5' over here, and 5' over here. It should edit a 3' too. So since this is the RNA primers, that means DNA polymerase can just go in and add the free 3'. You always have to remember the polarities. There's a free 3' over here. That means DNA polymerase can just add over here. This will be your new 3'. Same way over here, and same way over here. Alright, so now that, let's say it's done, then the next one is DNA polymerase 1. Remember, the difference between DNA polymerase 3 and DNA polymerase 1 is their function. DNA polymerase 3, its main function is to elongate the new strands. So we've done the elongation. DNA polymerase 1 is basically the one that replaces the RNA primer. So we have this RNA primer here. Basically what it will do is it will remove the primer and make it a DNA sequence. I'm just gonna make it blue so that you kind of have the reference from it that it was removed. So that's where DNA polymerase 1 does its job. It's basically, it just removes the primer. Same way on the Okazaki fragments or on the lagging strand, it will just remove each one of the RNA primers and make it into a DNA. Right? So we're almost done. Last one is the ligase. I think I forgot what to say the function of the ligase, so I'm just gonna add it here. Basically, ligase, it seals the gaps. Seals the gap between Okazaki fragments. And one way for me to remember, gaps starts with a G, and ligase has a G on it. So I guess if it's better for you to just memorize it, then do so. But this is how I remember it. Ligase seals the gaps between Okazaki fragments. So if we apply it to this diagram, these are Okazaki fragments, right? You see how it's not continuous, unlike this one. This is a leading strand, this is your lagging strand, and it's composed of Okazaki fragments. Basically, ligase will come in. I'm gonna make this L, and it will just basically like fix the phosphate backbones between 5' to 3'. So it will just fix this and make it one long strand instead. Same right here. And then if there's another Okazaki fragment here, it will fix it there. So yeah, now you have one continuous strand, and in the end it will just be one double-stranded DNA, one whole replicated DNA strand. So yeah, I hope this video helped, and if you have any questions, feel free to email us and ask us questions in class. I hope this helps, and thank you for watching.